Hello everyone, welcome to Biotech Notebook. In this video, we are going to see what is enzyme inhibition and types of enzyme inhibition and the difference between two types of enzyme inhibition, that is reversible inhibition and irreversible inhibition. Okay, so first let's see what is enzyme inhibition. Okay, as we know that enzymes are proteins, okay, which act as biological catalyst. So these biological catalyst is used to increase the rate of biochemical reactions. Okay, and these enzymes are very specific to its substrate. Okay, your enzyme catalyst reaction is enzyme plus substrate gives enzyme substrate complex, and this enzyme substrate complex dissociates into enzyme and product. Okay, so the substrate goes and bind with the active site of the enzyme. Okay, and it forms enzyme substrate complex. Then this enzyme substrate complex dissociates into enzyme and product. Now this reaction is regulated or inhibited by the enzyme inhibitors. So enzyme inhibitors are mainly used to regulate this enzyme catalyzed reaction, or it may be used to inhibit this reaction. Okay, so enzyme inhibitors are used to regulate or inhibit the enzyme catalyzed reaction. Okay, let's see about the inhibitors. Inhibitors are substances which bind to the enzyme and it reduces the activity of the enzyme. Okay, inhibitors are denoted by I, so which goes and bind with the enzyme and it reduces the activity of the enzyme, thereby it decreases the rate of enzyme catalyzed reaction. Okay, next, the mechanism of enzyme inhibition reaction. In enzyme inhibition, there are different types of enzyme inhibition. So one type of enzyme inhibition, your inhibitor is closely resembles the original substrate. Okay, so this inhibitor is known as substrate analog. So this inhibitor goes and bind at the active site of the enzyme and it prevents the binding of the specific substrate to the enzyme, so thereby it decreases the activity of enzyme and decreases the rate of enzyme catalyzed reaction. In another type of enzyme inhibition, your inhibitor goes and bind with the enzyme substrate complex. Okay, and it will form enzyme inhibition substrate complex. So it inhibits the product formation from the substrate. Okay, example for enzyme inhibition reaction is the treatment for methanol poisoning by ethanol. Okay, methanol is toxic to our body when it is converted to formaldehyde by the enzyme alcohol dehydrogenase. Okay, so here the substrate is methanol and your enzyme is alcohol dehydrogenase and the product is formaldehyde. Okay, so to treat this methanol poisoning, we have to stop the product formation, that is the toxic product formaldehyde. So in treatment of methanol poisoning, we are using ethanol as an inhibitor. So this inhibitor ethanol compete with methanol and it goes and bind with the enzyme alcohol dehydrogenase and it prevents the formation of product formaldehyde. Okay, so ethanol can be used in the treatment of methanol poisoning. Next, we will see the types of enzyme inhibition. There are two types of enzyme inhibition. One is reversible enzyme inhibition and another one is irreversible enzyme inhibition. Okay, next, we will see the difference between reversible inhibition and irreversible inhibition. Okay, in reversible inhibition reaction, your inhibitor goes and binds to the enzyme in a reversible manner. As an irreversible reaction, your inhibitor goes and binds with the enzyme in a irreversible manner. Okay, so in reversible inhibition, the binding is in a reversible manner. That means there will be a non-covalent bond formation between the enzyme and inhibitor. Okay, but whereas in irreversible manner, there will be a covalent bond formation. That means there will be a stable complex formation in between the enzyme and irreversible inhibitor. Okay, so then we can remove this inhibitor. So this inhibitor is bound with enzyme by non-covalent bond formation. Okay, so it is loosely attached with the enzyme. So we can remove simply by dialysis process or simple dilution. Okay, Whereas in irreversible inhibition, there will be a strong bond formation, that is covalent bond formation between your enzyme and inhibitor. So we cannot remove the inhibitor from the enzyme, but sometimes you can reverse the reaction by using chelating agents like EDTA, that is ethylene diamine tetraacetic acid and citrate. Okay, these two may be used to reverse the irreversible enzyme inhibition reaction. Okay, some example for irreversible inhibitors are heavy metals like lead, mercury and cadmium. Okay, after removal of your enzyme, okay, by simple dilution process or dialysis process, in reversible inhibition reaction, the enzyme retain its full enzymatic activity. Okay, whereas in irreversible inhibition reaction, your enzyme lost its enzyme activity. So in reversible inhibition reaction, your enzyme binds to an enzyme in a reversible manner, that is in non-covalent bond formation between the enzyme and your inhibitor. Then we can remove the enzyme from the inhibitor by simple dialysis process or simple dilution process. And after removal of your enzyme from the inhibitor, the enzyme retains its full enzymatic activity. In irreversible inhibition, your inhibitor goes and binds with the enzyme in a irreversible manner, and there will be a covalent bond formation. There is a stable complex formation between your enzyme and inhibitor. It is difficult to remove the inhibitor once it binds with the enzyme, but sometimes chelating agents like EDTA and citrate may be used to reverse this reaction. Okay, so irreversible inhibition reaction, your enzyme lost its enzymatic activity. Then there are three types of reversible enzyme inhibition. One is competitive, then non-competitive and competitive. Okay, the irreversible enzyme inhibition, there are also three types of enzyme inhibition. One is group specific reagents, then reactive substrate analog and suicide inhibitors. Okay, in the next video we will see in detail about the types of reversible inhibition and irreversible inhibition. Thank you for watching my videos. Please do subscribe and share if you like my videos.